It's Charles Dorman, who's continuing our theme of regulation of DNA transcription. Thanks very much, Prof.
Okay. So this is a physiology talk, and the story is going to be approached from the perspective of bacterial physiology. I'm at a microbiology institute, and we are particularly interested in the processes which are used by bacteria to control gene expression, in particular at a global level. And at the moment in our field, we're faced with this plethora of new information that's derived from bacterial genome sequencing programs. Thousands of microbial genomes are now available. This landmark of 1,000 genomes was passed about 2 years ago. And people struggling to make sense of all this information look into these catalogs and try to draw comparisons between the new genome sequences and those of what we call model organisms. So there's a lot of pattern recognition going on. We think that it's going to be very important to consider gene expression patterns in this context. And to that end, it's worth comparing some very well-characterized model organisms like, for example, E. coli and Salmonella enterica, which have been studied in great detail for decades. We have at least 60 years or so of detailed molecular analysis to draw on in the cases of these two organisms, and to test some of the assumptions that are made about the extrapolation of insights from model organisms to less well-characterized bacteria. So these two bacteria separated from the last common ancestor about 100 million years ago. And although they've gone their own ways, they have retained a great deal of similarity in terms of their genome structure. So the genomes continue to be similar in size. They have a remarkably similar gene content, and the order of the genes is highly conserved as well, as are many of the key regulatory features. And yet, it's possible to take out individual open reading frames that are absolutely identical, completely conserved between these species, and demonstrate that they're regulated in their bacterial hosts in distinct ways. So for example, you can pull out a gene with identical open reading frames and measure its transcription in response to some environmental factors that interest you, for example, changing the temperature, changing the pH and so forth, the presence or absence of oxygen. And what you may find is that in some cases you can see distinct levels of response on the part of this highly conserved gene to the same signal, and in other cases the response is pretty well identical. And what's the explanation for this? And does this tell us something about the role of this gene in the operation of the physiological pathways of these organisms? So you probably start by looking at the transcription signals, and often they're quite well conserved as well. But you do begin to see differences when you explore further upstream, looking for binding sites for regulators that act in trans at these loci. So in this example here, there are two binding sites for two regulators that are used by both of these genes, but in distinct ways. And there's a binding site here for a regulator which is specific to this gene. This one either has lost or has never had at all. So just to give you some real-world examples of this, I had a postdoc in the lab, Andrew Cameron, do an analysis of this sort for some important genes that are involved in running many of the important pathways of the cell. And this is the situation upstream of a gene called the FIS gene, which Andy Travers has worked on in great detail in his lab, particularly in the E. coli system. And it's a comparison between the situation in E. coli and Salmonella. And what you can see is that once you get past the transcription initiation point here, you can start to see differences between the E. coli and the Salmonella situation. So these red boxes here represent binding sites for the FIS protein. So this gene is autoregulated. But the positions of these sites are sometimes conserved and sometimes not when you compare across species. The integration host factor, which we heard about in the previous talk, is also binding here, and that site is conserved. But you have sites for this CRP protein, which are found in the E. coli example, that are absent from the Salmonella example. So these genes have started to acquire expression patterns that are distinct, and we think we can explain this based on the presence and absence of these types of sites. In the case of DNA topoisomerase 1, the top A gene, which encodes this important regulator of DNA topology, we have preservation of the transcription initiation points, but differences in the positions and numbers of binding sites for the FIS regulator. Now this matters in terms of these genes themselves, but it also matters for many other genes, because in both cases, these genes lie upstream in regulatory hierarchies. So there's going to be a cascade of effects through the hierarchy. And we think this is an important factor when one comes to consider the basis of bacterial speciation and what it is that distinguishes one bacterial species from another. Now, all of us in whatever field we work in are to some extent prisoners of the history of that field. And in my field, we have been very heavily influenced indeed due to the fact that some of the early stories in gene regulation involved proteins which regulate transcription by binding to specific sequences, operator type sequences, and either repressing or activating transcription. And a great deal of energy has been devoted to understanding these sorts of relationships. And more recently, there's been an industry has developed in the field in which the role of regulatory RNAs, either working in trans or in cis, in controlling gene expression, primarily at the post-transcriptional level, has been emerging. It's also important to consider the Cinderella molecule in gene regulation, and that's DNA itself. We rarely come across DNA in discussions of gene regulation other than as a template upon which the regulators act. And what this talk will hopefully try to do is to activate some interest in considering DNA itself as an active player in the process of gene regulation at a global level. Now, to do this, it would be useful if the structure of DNA were variable, and if those variations could be programmed in some way that were physiologically useful for the cell. And then we could integrate that variation with the more traditional ways of looking at gene regulation. So in this model then, we have a hierarchy. And the hierarchy consists first of all of the DNA template and its topological state, which of course, as you know, is a manifestation of its energy content and its likelihood of engaging in processes such as transcription or replication, transposition, other forms of recombination. And at the top of this cartoon here, we have an open complex here, which is a place where transcription may start. So in addition to the DNA then, we have to consider these nucleoid-associated proteins. Now there's a prejudice in the field that bacteria don't have chromatin. That's probably an oversimplification. Bacteria do have structures that approximate to chromatin, and in this structure you have a role for these nucleoid-associated proteins. They're involved in organizing the superstructure of the nucleoid. They're also involved at a local level in regulating transcription. And in some cases, they do this by constraining supercoils and by otherwise manipulating the local shape of DNA. And then we have the conventional transcription factors. These are the founders, I guess, of the gene regulation field in bacterial circles. And these then cooperate with the state of the DNA, the presence or absence of the nucleoid-associated proteins, finally to recruit RNA polymerase to the promoter and activate transcription or to otherwise modulate transcription. So this is a hierarchy then with DNA having a big say in what goes on, the nucleoid-associated proteins also making a major contribution, and then these transcription factors making their contribution very much at a local and targeted level. And RNA polymerase then has to operate on this landscape. So over the development of a bacterial culture, we see a number of distinguishable phases. And these sometimes get ignored, particularly in the biochemistry community, who simply use bacteria as factories to produce their favorite molecules. We're very sensitive to the fact that the bacterium experiences a variety of physiological states as it moves from lag phase to exponential growth, and then into what we call stationary phase, where there is a cessation of growth. And over this growth curve, we see the comings and goings of major players which are involved in running the global regulatory picture in the cell. So for example, there is a very important nucleoid-associated protein called the HNS protein, which is present at a constant level throughout growth. That's constant with respect to the number of chromosomes in the cell. There's an attempt made by the cell to match HNS quantity to chromosome quantity. But HNS is pretty much around all the time, and the cell has to deal with that due to the impact that it has on global transcription. In young cells, going from lag phase into exponential growth, there is a burst of expression of the FIS protein, which I've already mentioned to you. And this is a key player in the upregulation of the translational machinery of the cell. So in order to build cells, you need to make that machinery, and the FIS protein is critical for that development. And it has tended to be associated with this early stage in exponential growth. But this turns out to be a highly simplistic picture. We can extend, for example, the expression of the FIS protein well into stationary phase by manipulating the growth conditions. At the other end of the spectrum, then, you have a role for this DPS protein, which is associated with elderly bacteria, bacteria that are getting into stationary phase. And it has a key role to play in protecting the DNA, although its role in influencing gene expression is very poorly understood indeed, and it may not actually have a major role in that process. The integration host factor, which Craig has just mentioned, is present throughout growth, but its concentration peaks again at another transition. This was the transition from exponential growth into stationary phase. And at that stage, you also see the appearance in maximal numbers of the LRP, or leucine responsive regulatory protein. So there are up to 12 nucleoid-associated proteins in bacteria like Salmonella and E. coli. And I'm not going to go through all of them, but I will finally mention the HU protein, because it's the best conserved across all bacterial species as far as we can see. Even mycoplasmas, which only have one, have this protein, HU. And it has a little bit of variety associated with it in that it comes in 3 flavors. It's a protein that forms homodimers. This is the alpha-2 homodimer, which is associated with young cells, the beta-2 homodimer, which is associated with elderly cells. And most of the time you have this alpha-beta heterodimer. And these 3 forms of the protein do 3 different jobs in the cell because they have different properties and are able to do different things with DNA. So it's a very dynamic situation, which is made even more dynamic when you consider the fact that the DNA upon which these proteins wish to act is itself dynamic. So in young cells where energy levels, these are the lag phase cells, energy levels are low, what you find is that the DNA is relatively relaxed in those cells, and it becomes more negatively supercoiled as the metabolic pathways in the cells crank up, and you see a shift upwards in the ratio of ATP to ADP, powering up gyrase, which is then available to transfer energy to the DNA by negatively supercoiling it. And as the bacteria run out of gas, getting into stationary phase, you see a relaxation of the DNA again. So there's this moving landscape which all of these proteins which are coming and going have to act on. So it's a very dynamic situation, but it's not a complicated situation in that you can actually understand this if you pay attention to it. Now, to make the story flow, it's usually best to talk about a specific case. And the case I want to talk about is the case of Salmonella typhimurium, which is a facultative intracellular pathogen. That means it can, if it wants to, invade host cells. It doesn't have to do that. It's not an obligate intracellular pathogen, but it can do that. And it can, the form of Salmonella that we work with, Salmonella enterica, is a serovar called typhimurium, which means mouse typhoid. So it causes a typhoidal disease in mice, which is a very good proxy for the same disease in humans. And it's been studied intensively for that reason. And the bacteria are acquired through ingestion of contaminated water or food, and they undergo a journey through the digestive system of the host, which exposes the bacteria to a series of problems in the form of environmental hurdles that they have to overcome in order to move on to the next stage. And this is solved by the bacteria through gene regulation. Okay, so the primary contributors then to the solution of the problems concern what we call type 3 secretion systems. These are machines that look a little bit like this. They are actually molecular syringes, and they're deployed on the surface of the bacterial cell at the right time and in the right place to inject into the human or murine cells proteins which manipulate the signaling pathways in those cells, so that in the case of the gut epithelium, and this is in the small intestine, the bacteria can cause themselves to be ingested by these cells, which would normally not exhibit this type of behavior. Then they can go on to infect defense cells of the host. These are macrophage. They're there to kill bacterial invaders, but the Salmonella have the ability to survive in these vacuoles in macrophage because they can modify those vacuoles using a type 3 secretion system. So there are 2 to think about. The SPY1 type 3 secretion system is required for the invasion initially, and the SPY2 type 3 secretion system is required for the vacuolar survival. And these sets of genes which code for these type 3 secretion systems, and the effector proteins that are pumped through them are reciprocally regulated. So early on we have high-level expression of the SPY1 set, and then later on at this stage we have upregulation of the SPY2 set, which might imply some kind of communication between them. So the possession then of these genes is one of the things that distinguishes Salmonella from E. coli. And we know where these genes live, and they are present in large blocks of DNA, and these blocks of DNA have been imported horizontally from an unknown source. We know that they're not Salmonella DNA because they have a structure which is distinct from the core genome of Salmonella. They are highly AT-rich, and this produces some interesting problems for the bacterial cell in terms of taming these genes and using them in ways that benefit the bacterium. So AT-rich blocks of DNA in the chromosome, and we call these pathogenicity islands. So SPY means Salmonella pathogenicity island. First thing to say is that these represent magnets for the HNS protein. HNS is this nucleoid-associated protein that's around all the time. And the HNS protein targets DNA, which is high in AT. And this is a chromatin immunoprecipitation experiment that was carried out by another postdoc, Shane Dillon. And what he shows here is a distribution of AT richness along a portion of the Salmonella chromosome. And you can see in the chip data a very nice correspondence between these peaks for HNS binding and the peaks of AT richness here. And the SPY2 gene set is in here. And SSRA-B, this is the master regulator of the SPY2 gene set. So HNS is very important to Salmonella in downregulating transcription of both SPY1 and SPY2, because we see the same pattern over there. So what's HNS all about? Well, in essence, what this thing is a bidentate DNA binding protein. It's a dimer, and it has an N-terminal domain that's involved in dimer formation. It has a C-terminal domain that's involved in DNA binding. And it's able to interact with DNA roughly in two ways. In one sense, it can form a nucleation at a nucleation site, a polymer along the DNA, which is capable of excluding RNA polymerase from promoters. And in bacterial cells, promoters are generally AT-rich. So HNS is a fabulous mechanism for downregulating transcription globally. The other thing it can do is to form bridges between different parts of the genome, or between different DNA molecules. Inter and intramolecular events are possible. And in some cases, you can get a blockading of RNA polymerase at a promoter from which it cannot now escape. So given that bacteria have to live with this thing, they have devised or evolved a whole series of mechanisms for overcoming HNS repression. And I'll just give you a few examples. So this is the repression complex, and I've chosen the bridged complex here. It doesn't have to be the bridge complex. It could also be the polymerization complex. And what we find in some cases is that altering the topology of the DNA at the bridge itself is sufficient to compromise the structure of that bridge and allow RNA polymerase to escape. In other cases, what we find is that you can get proteins in that can form blockades to polymerization of HNS along the DNA and can protect downstream promoters. In this case here, we have proteins that are involved in remodeling the bridge, causing the HNS protein to be displaced from the DNA. In more subtle cases, some of these same proteins are able to remodel the bridge in such a way that it is no longer capable of containing RNA polymerase, and you can get transcription out of this complex. In some other cases, we have traditional conventional transcription factors which are simultaneously able to recruit RNA polymerase and displace the HNS protein. In this case here, we have the production of a tiny protein which approximates to the oligomerization domain of the HNS protein but lacks the DNA binding domain. These insinuate themselves into bridges and weaken their structure, allowing them to be broken. And then we have various mechanisms by which you can control locally the supply of HNS to the target sequences, resulting in weaker bridges. And then finally, you can dope the HNS population with analogs, proteins which are similar to HNS, but which retarget it to sequences that it would not normally bind to and away from their natural targets. So given that HNS is a fact of life for Salmonella, as it is for many gram-negative bacteria, it's not surprising that so many solutions to the HNS problem have evolved. And this is not a complete listing by any means. Now going back to SPY1 and SPY2, I've told you that these genes get decorated by the HNS protein, leading to their downregulation. But if they're going to do Salmonella any good in the context of infection, they have to become upregulated at some point. And this happens by an interesting combination of players that have been imported into the Salmonella along with these horizontally acquired genes, and other players that lived in the bacterial cell previously. They're part of the ancestral or core genome. And that's an interesting example of integration of regulation involving imported genes and pre-existing regulators. And it's a very important topic in our field, trying to understand how that can be brought about quickly in order to facilitate utilization of imported genes. Because horizontal gene transfer is going on all the time at high frequency through 3 mechanisms. It's going on in this room right now. And we have DNA being imported into bacterial cells in our microbiomes through either phase transduction or transformation or through conjugation. And cells have to deal with that. Because cells, these bacterial cells, take exams constantly from the environment. And if they fail those exams just once, they're out of the game. So what we see here is that the SPY2 gene set is upregulated by this SSRAB 2-component regulatory system, in which one of the players is a kinase associated with the cell surface, picking up environmental signals that tells the system that it's your turn to be switched on. It's also picking up signals from a system that's encoded by the core genome called NZ-on-board, which is also giving information to SPY2 that it's in the right site in the host in order to be switched on. And the SPY1 gene set has its own set of regulators, one of which happens to be this on-board protein, part of the core genome contribution. There's also communication going on between SPY1 and SPY2. They have to talk to each other so they know what each other are doing at any one time during this process of infection, invasion, and survival in macrophage. Okay, so going back then to this representation of the landscape, what I want to switch to talking about now is this variation in DNA topology, but doing this conversation in the context of the Salmonella infection system. In order to be useful in terms of upregulating or downregulating genes that are involved in environmental adaptation, it will be useful if DNA topology in bacterial cells were environmentally responsive. And it turns out that it is. These are sets of gels actually ran in the late 1980s, so we've known about this for a long time. Basically, if you abuse bacterial cells in a variety of ways by changing their environments, one of the readouts for that is a change in the linking number of reporter plasmids, which tells you things that are going on the chromosome. And this has been followed up in detail, looking into genes in the chromosome that are topologically sensitive. So environmental factors then, things like osmotic stress, the presence and absence of oxygen, changes in pH, and so on and so forth, have indirectly the ability to manipulate the degree of supercoiling of genomic DNA in bacterial cells. And an obvious, to us anyway, corollary from this is that this could be utilized by a pathogen during the infection process. So the sort of stresses that these bacterial cells are dealing with in routine external environments are also faced by pathogens during infection. So therefore, it's likely that they could exploit this as a mechanism for influencing pathogen-specific gene expression. I can't go on to talk about that without mentioning once again that the FIS protein, because the FIS protein is an intimate player in the setting of supercoiling set points in bacteria like Salmonella. It has a role to play in the transcriptional regulation of the genes that make the major topoisomerases. It also has a role to play in the modulation of the supercoiling in DNA because it can protect DNA from over-supercoiling or over-relaxation by these topoisomerases. At least that's been shown in vitro. So the FIS protein is likely to be important in terms of topologically buffering the system in addition to the major inputs of topoisomerase 1 and DNA gyrase. A number of years ago, during a survey of FIS-dependent gene expression in Salmonella, Arlene Kelly discovered that the entire set of FIS genes in Salmonella typhimurium is FIS-dependent for upregulation. So this is the master regulator of the SPY2 gene set, and it is dependent upon the FIS protein for expression. Just to show that that matters in terms of infection, if you have the SSRA promoter driving the GFP, This is a green fluorescent protein marker that we use to follow gene expression while the bacteria are inside mammalian cells. The induction of that system is undermined if you don't have the FIS protein present in the cell. And that actually has downstream consequences for the survival of the bacteria. So in the macrophage, over time, what you see is that the salmonella that don't have the FIS protein do less well, and the macrophage can now do their job, which is to kill these bacteria. So we were curious then about the topological state of the genome in salmonella while it's in the vacuole inside the macrophage. And another postdoc in the lab, Tycho Cronin, investigated this. So essentially the idea is to go into the macrophage, find the vacuoles, find the salmonella, pull out reporter plasmid DNA and see what state it's in. And what Tycho discovered was that over a period of time that the bacteria spent in the macrophage, the plasmids became progressively relaxed, which is a situation that's easy to replicate in vitro using gyrase inhibiting antibiotics. And what you see here is that, again, this SSRA promoter is positively responding to progressive inhibition of DNA gyrase with novobiocin. And that is true whether the gene is studied in a plasmid situation or in its native location in the chromosome. You see this is RNA being expressed from the SSRA gene as the DNA is relaxed due to gyrase inhibition. A complication arose when, probably not surprisingly, it was announced that actually E. coli, which is this close relative of salmonella, and salmonella, maintained their DNA at distinct set points under identical growth conditions. A paper from Pat Higgins, published a few years ago, reported that a form of GRB, a mutant form of GRB, which was simply temperature sensitive in salmonella, was lethal when moved into E. coli, and that a reported plasmid had a 15% difference in negative superhelicity in E. coli compared to salmonella. So put superficially, salmonella is a more relaxed organism than E. coli. And that may mean something in terms of things like horizontal gene transfer and the operation of the global regulatory systems in salmonella. So we had a quick look at this, and this is work from two postdocs, Andy Cameron and Dan Stobel, comparing the situation with regard to plasmid superhelicity in E. coli and salmonella cells. So these are chloroquine ladders, the topoisomer ladders on agarose gels containing chloroquine. This is the effect of inhibition of DNA gyrase using a low concentration of novobiocin on this distribution. So the box plots allow you to follow this more easily on the screen. So what you see in salmonella is that novobiocin treatment causes the expected relaxation. And in a FIS mutant, you see a similar pattern, although it's not as exaggerated as it is in the presence of FIS. E. coli responds better to the treatment, and there isn't really much difference between the FIS mutant and the wild type under these conditions. And then looking at the situation in respect to some environmental signals that we know are important to salmonella during infection. And this is an attempt to replicate osmotic stress using sodium chloride. These are distributions of topoisomers from E. coli and salmonella wild type, and from E. coli and salmonella where the FIS gene has been deleted. And what you see is a very complex pattern indeed. But basically what happens in E. coli wild type is you see a strong shift in supercoiling towards the more negatively supercoiled end of the distribution. And that also happens in the FIS mutant. The salmonella bacteria, on the other hand, show a much more muted or even no response to this type of stress. Interestingly, if we change the growth conditions by one more parameter, so this is a standard set of growth conditions of high aeration. If we use low aeration conditions, which are the sorts of conditions that obtain during infection, for example, of the small intestine epithelium, what you find now is that E. coli and salmonella become indistinguishable both for the wild type and for the FIS mutant. So we're able to manipulate the global superhelicity in these species genetically and environmentally. We then did the horizontal gene transfer experiment ourselves. So we took the Salmonella enterica SSRA GFP system that I've already mentioned to you and studied it in the wild type cell as a function of increasing osmolarity and DNA relaxation arranged by gyrase inhibition. And you can see the kinetics of induction here for 0 NaCl, which is the type of low osmolarity environment that you experience in the vacuole, and DNA relaxation. This gives the optimal induction pattern for the native gene in its native location. If you switch this gene into E. coli, it turns out that these are actually the worst set of conditions for its upregulation. But if you get rid of the FIS protein from both bacteria, you can now bring these into line with each other. So again, manipulating the bacteria genetically and environmentally can perturb their responses to these environmental signals at the level of this topological response. And finally, I just wanted to look at briefly the issue of the decoration of the DNA by global regulatory proteins as a function of the supercoiled state of the DNA. And what you need to focus on in this rather crowded slide are 4 data sets here. For promoters, SSRA, once again, the on-board promoter, this is this part of the core genome that is an important player in SPI1 and SPI2 regulation. The promoters of HIL-C and HIL-D, which are 2 of the major regulators of transcription in SPI1. And what we're looking at is the binding using chip analysis of the on-board protein, the HNS protein, and the FIS protein to these promoters in a supercoiled and in a relaxed state. And what you see is that for the HNS protein in all 4 cases, relaxing the DNA really makes no difference to the ability of the HNS protein to target and bind those DNA sequences. In the case of the on-board protein, the binding is enhanced by DNA relaxation, and in the case of the FIS protein, we see a reduction in the binding of the FIS protein due to DNA relaxation. So when we perturb DNA supercoiling in all these various ways, we shouldn't just think about the events in terms of formation of open complexes and things like this, transitions within the structure of the DNA itself. We should also consider what we're doing to the DNA in terms of making it more or less attractive to passing proteins which may want to bind to it. It isn't simply a matter of the presence or absence of particular target sequences, the structures matter as well. And this is particularly the case with proteins like ONPOR, HNS, and FIS, which have got very low sequence specificity in terms of their binding requirements. They're actually just looking for AT-rich DNA most of the time. Okay, so what I'm going to finish on then is fish out this ONPOR promoter, and this is an experiment I actually did specifically for Craig Benham, and you'll see why in just a minute. What we did was we looked at this promoter in vitro, looking at the decoration of the FIS promoter, looking at the decoration of the ONPOR promoter by the ONPOR protein at a variety of superhelical densities. And we also ran this promoter sequence through Craig's SID predictor. And there's some interesting information coming out here about the likelihood of deformation of this piece of DNA, and the fact that this coincides with the major binding site for the ONPOR promoter at its own, at the ONPOR protein at its own promoter. So maybe we'll get to discuss that at the coffee break. So to wrap up then, what I've tried to communicate to you this morning is that what we're dealing with in these bacterial systems is a highly dynamic situation. The textbooks and the review articles are full of statements about the comings and goings of these proteins and their properties and what they'll do to various collectives of genes. In the case of HNS, we're talking about up to 1,000 genes that are responsive to that protein. And people make predictions of what may happen and then extrapolate those to other species. What I'm trying to communicate to you is the fact that all of this stuff is provisional and it's conditional. And it all depends on things like how you handle the bacteria. And even though 2 species may have a lot of distinct features, in some instances you can bring them into alignment by judicious manipulation of them, simply environmentally or perhaps by a combination of genetic trickery and environmental perturbation. Okay, so I'll just end then by renaming some of the contributors. The postdocs, Andy Cameron, Shane Dillon, and Dan Stovall I've already referred to, as I have Tiger Cronin in terms of the development of these systems I've spoken about. Arlene and Orla were involved in some of the early work we did with the FIS system, and Science Foundation Ireland paid the bills. So with that, I'll stop, and if you have questions, I'll be pleased to answer them.
Questions for Charles?
So the regulators, the SRS regulators, that regulate the genes that were horizontally transferred. Could it be possible that they were also regulated from wherever they came by the same kinds of global regulators?
Can you repeat the question?
So the question is, it's a speculation about the biography, I suppose, of SSRA-B. And the question is, in wherever they came from, do they have a similar set of regulatory problems to solve and where they control it the same sort of way? It's entirely possible, but because we don't know where they came from, we can't address this. And it would be great to be able to answer this. The best we can do at the moment is try to recapitulate these horizontal transfer systems artificially, and that's being done.
All right, let's save the rest of our questions then for the coffee break.